All right, good morning. Here we go, and hopefully this one will work this time. Um, so when we look at the Rh blood group system, it's actually very simple, but the nomenclature makes it way more difficult than it needs to be. I'm going to skip over the objectives. Those are designed to kind of set and help you organize your notes and whatnot. So we're going to skip through those. And don't mind the noise in the background. It's my two giant dogs fighting. Um, but we want to take a look at history of the blood group system. So in 1940, Levine and Stetson actually figured out that the Rh system is actually connected to hemolytic disease of the fetus and newborn. So you'll see it interchanged sometimes in the documentation, HDN or HDFN. They are both the same thing, but we just decided to start clarifying it as HDFN, or hemolytic disease of the fetus and the newborn. Landsteiner and Wiener. I think it's actually pronounced Weiner, but I just have way more fun calling it Wiener because that's way cooler. So they found that anti-RH agglutinated 85% of human red blood cells, and then 15% didn't agglutinate. So this is where we started coming up with the Rh positive and Rh negative. And that's simple enough. Rh positive means you have the D antigen on your cells. Rh negative means you don't have the D antigen. So that's how that's connected. Sorry, I'm still slightly recovering from this cold. So you'll get a better audio, I think, on this one. So the anti-human or the human anti-Rh was similar to guinea pig and rabbit antibodies that were made when they were exposed to rhesus monkey RBCs, hence the name RH. Both antibodies were actually later on identified as different, but they just kept the Rh, which is like the first time in science that they didn't rename something, or they didn't name something over after a scientist. So when we actually got further down into investigating the rhesus antibody, it actually ended up being directed towards the LW antigen, or the LW family. So basically, our current theory that we have now is that there's two closely linked genes that control our expression of all the Rh antigens, and they are co-dominant alleles. So that's what we're operating now. So in this case, the RhD antigen will determine the expression of our D antigen. So the RhD gene says whether we are D positive or D negative, which means it says whether we are Rh positive or Rh negative. The RhCE gene will actually tell us, do we have big C, little c, big E, and little e? So all of these upper and lowercase are actually indicating antigens. So we will inherit a combination of the capital C, capital E, little e. And I know when you're first reading it, you're like, what? That just means they just use these letters to name the antigens that are on the red blood cells. That is all, believe it or not. So when we look at genetics and biochemistry, we had some other theories that were initially postulated. Our Fisher race, where the Rh antigens are really connected by closely linked loci. So big D, big C, big E, little e. And then the Wiener, where the Rh antigens are controlled by alleles at one gene locus. So what we did is we basically, to make it easier, combined things. So these different things that they listed in this chart, this looks kind of scary, but it's actually just telling you the equivalent. So we will typically see these. We will see the Fisher race nomenclature. We will use those for when we're identifying antibodies and how we're investigating them. Um, and you'll see a antigram real soon, and to kind of show you that, I'll post up a picture of one as well. But the numeric is basically um, they take number one, and that's the D. Number two, it's the C. It's just equivalent. You don't have to spend a lot of time memorizing these. These are typically what we're going to see as basically describing what cell is being utilized within the panel. And then other names that we'll see, obviously Rh positive, which you do know. And then you can see things like um, the cis or CE or F versions. And the ISBT number is actually really simple. The ISBT number is just a numerical value that they assign for the computer. It's just to make it easier when we're cataloging blood and basically making sure that the donors that are antigen typed appropriately in the computer, and the antibody that the patient has is done correctly in the computer. So the Fisher race basically is named as each gene expresses an antigen, and it's given the same letter as the italicized letter of the gene name. So the C gene produces the big C antigen. So the absence of the D antigen is written as little d or small d. And the order of these is usually DCE, but we can order them alphabetically on occasion. So when we look at the Wiener terminology, um, they term it as 8 alleles that exist at the single Rh gene locus. So R0, R1, R2, RZ, little r prime, little r double prime, little ry, and each will encode an agglutinogen, which is made of different factors that correlate with Rh antigens. So basically our R1 is our RH1, which is also our big D, big C, little e. We typically don't use that blue long-handed notation, and a lot of this Wiener terminology is starting to go away. Nobody likes it because it's very confusing to remember, and it's simple memorization, trying to figure out what is equivalent to the other. So how do we convert it? So basically, for example, if we look at this first one, your R0 here, R0 is basically corresponds to a patient having only the D antigen, because this little, only the D antigen, and then has little c and little e. So the little or smaller letters are considered like the recessive antigen, if you will. So the R1, it'll have big C, big D, little e. So basically what this means, all these letter combinations, for example, we'll take this one here. So this basically just means that this is your cell, and you have the big C attached to it, and you have the big E attached to it as an antigen. That's all it means. So don't kind of beat yourself up over this. All you need to know is the equivalent. So I do recommend just remembering this, only because I have seen when I took my certification exam, I got one question too many wrong in this area, and they gave me a bunch more. But this was something I just honestly I didn't put any time to learning into in school because I thought it was ridiculous. Like, we use the Fisher race right now. Why the hell am I trying to remember something that we don't even use anymore? But apparently it's still out there. A lot of the old school texts that learn that first use that. And what they do on the certification exam is they give you um, more questions in the area if you get like one too many wrong, because they really want to make sure that you understand that area. So be prepared for that. So this Rosenfield was that Rh antigens are designated by a number. So that one's pretty simple. 1 is D, 2 is C, 3 is E, 4 is little c, 5 is little e. So that's pretty simple. Your ISBT terminology. So basically, our 004 designation basically says this antigen is in the Rh system, and then the other numbers correlate with the Rosenfield system. So it literally is as simply as 004002 is our code for the C antigen. And these will change depending on what blood group they are in and what antigen it is. So it's basically just an attempt to standardize things like that. It's actually that simple. So it's just computer programming. So our genotype. So when we look at the phenotype, this is a result of the reaction between red cells and anti-sera. So the genotype is the genetic makeup, and we can actually predict the phenotype if we look, or we can predict the genotype utilizing the phenotype and the race of the patient. So we really only could do molecular testing, within a family to really truly do the genotype. So we can predict it knowing the race of an individual and some of our common phenotypes. So we have different phenotypes that are more common in Caucasians and different that are uh, common in Black population. So you can see over here where we have the highest to lowest frequency. And this will be things, so our most common is our R1, if we're Caucasian, and the R0 if we're Black. And then we do have some mix of races because let's face it, it's America now, and nowadays there's less stigma in mixed race marriages and mixed race babies. So this is an example of a chart that you can help predict your genotype. So typically we try and do a lab that has this where we antigen type ourselves to figure out our genotype. So I'm going to talk with Professor P, see what you have for anti-sera, because I think it'll be cool because then you can predict your genotype. So basically, this plus means there is reactivity, and the negative means there none. So, for example, I'm a negative, so I'm going to be somewhere in these down here because I would not react with anti-D because there is no D antigen on my cells. So I would be one of these. So chances are this is going to be me because this is the most common in a white race. When we did this with my students, I think we had someone that was down in this area here where they had the little c, big E, and little e phenotype. So it was really cool. So hopefully we will have some anti-sera so you guys can antigen type yourselves, which would be pretty awesome. And then you can predict your own genotype. So the D antigen itself is pretty immunogenic. It's the most immunogenic in the Rh system, and the Rh system itself is pretty immunogenic. So D negative patients should receive D negative blood, and basically, if we're D negative and we receive D positive blood, we can produce an anti-D antibody in response. And you can see how we have some phenotypes that have a lot of D antigen and some that have less D antigen. So we will test patients for weak D, and these are red cells that will only react in the indirect antiglobulin test or in the IgG phase, if you will. We call these weak D antigen. Most of our monoclonal reagents now detect our weak Ds without using the full IgG phase, but we can sometimes miss it. So all donors are required to have weak D testing. Most of our patients, we do not have to, unless they're babies. So this is more common within the Black population, and it comes from a weaker form of the D antigen, and it often comes in the form of the little c, big D, little e haplotype genetically. So that's the typical one that we see, and we usually use that IgG phase. We are considered weak D positive, and they do get D positive blood. So one of the other things that we can see is if a D antigen appears next to a C antigen, it can actually appear a little weaker because basically it gets in the way. So we can see that interfering with the D activity. The CE gene is paired with CDE or little CD, big DE, or your R1 and R0. So that gets that would be how we would potentially see a weak D as well. And they consider these patients weak D, and we give them D positive blood. So we can also see things that are called a partial D. So we basically we're typing as D positive, but we don't have the D antigen in its entirety. So what ends up happening is we've got part of it, but when we're exposed to part of it, because we are technically, we don't have the whole thing. We type as D positive because we do react with the reagent, but when we receive, and this is if I'm speaking as if I were a patient that had partial D, when we would receive D positive blood, we could potentially make an antibody against the full D antigen part that we don't have. So these partial Ds will react with monoclonal reagent. So we would suspect this if we had basically the patient is typing as D positive and makes anti-D, and then it doesn't react in the autocontrol. So this is where we would think it would be a partial D. So AABB requires that all of our donors be tested for weak D just in case once they're initially non-reactive, and we don't need to do it on a recipient. Patients will be classified as D negative and transfused with D negative blood, so it's not the end of the world. So we also see things called what's called compound antigens. They're called cis product antigens. So two genes inherited on the same chromosome can form additional products. So the most common one that we see is the little c and little e antigens will cause a little f to be inherited along with them, and that is all. We also have G antigens, so almost all genes that code for C or D, or big C, big D antigen will code for G. So anti-G will look like anti-C or anti-D. So if a patient is negative for big D and or big C and gets a D positive or C positive blood, she can also produce anti-G in addition. So if anti-G is present, what we would do is give big D and big C antigen typed negative blood, because you need C and D to have G. So some of the unusual phenotypes, now these you could like essentially never encounter. They're pretty rare. So we have a D deletion, an RH null, and an RH mod. So basically, we're starting to see missing reactions within our RH antigen typing. In a D deletion, there's a stronger D antigen activity, so we'd have to transfuse only D deleted cells. We would get these from a rare donor bank. And then Rh null has no Rh antigen coming from that regulator gene or an amorph gene. And then the Rh mod is close to the Rh null, but it's controlled by the RHAG gene. So Rh antibody is usually red cell stimulated. Most are immunoglobulin G or IgG. We will see the agglutination in the indirect antiglobulin phase, and potentiators are useful for identification. So typically speaking, we use lists in the laboratory. Can't forget to add that. It's important because what happens is if you could miss a weak antibody expression if you don't add the potentiator to get those cells closer together. So antibodies to big C, big E, and little e will react stronger to homozygous antigens. So basically, this means that a homozygous antigen is if we inherit, um, for example, a big C and a little c. Okay? Because this means if we've got the big C and no little c, this means that on our red cell, we have a double dose of C antigen. So it will react more. Okay? So that double dose will cover the antigen versus if it was a big C. So basically, if the patient has big C and no little c, okay, um, well, sorry, I wrote this out. You shouldn't. So if a patient has big C, right, and basically only has two copies of this, okay? So remember, we inherit and pair. If we only have the big C present, that means that this big C is going to be doubly on the cell. It'll take up the space that a little c antigen normally would. So if it had big C and little c, it would have a combination and less of it. So the double dose tends to react stronger. And we'll talk more about this as we move forward too. And I'll give you a quick little video example of dosage. So Rh antibody does not activate complement. And one of the things that's key within the Rh family is big E negative and little c negative blood is usually given when an anti-E antibody is identified. Because a lot of the times anti-little c is seen together with an anti-big E antibody. So we will screen our red cells, our donor cells, to give the patient big E antigen and little c antigen negative products. And we do this all the time. This is a rule in the blood bank. So why do we care about the Rh antigen? Well, we can see transfusion reactions because if they're not exposed to the Rh, it can go unpredictable, undetectable for years. But then once they're exposed to Rh positive blood, it can become a transfusion reaction and that antibody activity go up a lot higher. So we will give antigen negative red cells if we have any type of Rh antibody. And this is not just D. This is D, big C, big E, little c, little e. The Rh blood group can also cause hemolytic disease of the newborn or hemolytic disease of the fetus in newborn. This can occur when a mom is Rh negative and the baby is Rh positive. We can form antibodies upon first exposure to a fetal maternal hemorrhage or any type of bleed during the first pregnancy. And what will happen is this antibody can destroy fetal red cells in subsequent pregnancies. So if this patient is not protected, forms an Rh antibody or an anti-D, the future pregnancies can either be anywhere from the baby is born with hemolytic disease of the fetus and newborn, or it's a stillbirth or a spontaneous abortion, miscarriage, anything like that. So what we started doing was an Rh immune globulin. And this is basically human-derived anti-D, and they give it as an injection at 28 weeks. It hangs around for a couple months, and it protects the mom until after birth. And we actually will pick it up when we do a type and screen on a patient when they come in to deliver. We can see the Rh immune globulin reacting, and we have to identify. We call it a passive D or a passive anti-D because we've given it to them. So if the baby is born and the cord blood is tested and the baby is Rh positive, then the mom will get another dose. This will be at least one dose. They will do a fetal screen, and they'll actually screen for fetal blood, and could potentially have to do staining if that fetal bleed screen is positive. They have to do what's called a Kleihauer-Becky stain, which stains hemoglobin F. And they will do a calculation to see how many doses of Rh immune globulin that patient would need. And when we talk about the fetal maternal medicine later on in the course, we will go over that. Um, so the Rh immune globulin itself was actually initially formed because they discovered, and this gentleman actually recently donated his last unit, but he had gotten transfused and had this random antibody floating around, and that's where they call it the ROGAM now. So it's pretty cool. You can Google it. So the last blood group that's considered part of the Rh because it's similar is the LW blood group. They're similar to the Rh antigens, but they're not actually genetically related. Remember, we started discovering this and we started experimenting with rabbit and guinea pig antibodies against that rhesus monkey cells. So the anti-LW antibody can react with the D positive and D negative weak cells, and these will include your LWA, LWB, and LW. So the most common phenotype is LWA positive, B negative. This is very uncommon to see this causing a problem. So it's mentioned because you do need to know it exists, but we don't see them that often. This could fall under like a high frequency or low freak antibody if we're detecting something and we can't see what it is. It's reacting, but it's not any of the common antibodies. So that's Rh blood group in a nutshell.